Franz van der Vosse is not able to come, and Salama agreed shortly in presenting his work. Although it does not really totally fit into the session, I'm really grateful that you've been able to on this short notice give us information about your work.
Laser pointer and advance. Slide advance is here. Thank you. Well, thank you very much for the invitation, and Nick for organizing this wonderful meeting, and all of you, the audience, most of all. So, as you heard in the previous talk about our, cardiovascular diseases, it's somewhat related in that sex plays a major role in differences between men and women in terms of cardiovascular disease. And men tend to be much more vulnerable to cardiovascular disease than women. But in terms of electrical problems, things are reversed. So what I'm going to try is to give you a little bit of background on phenotype between men and women in terms of arrhythmogenic phenotype in the long QT syndrome. And here I'm lucky because Bianca just presented a very strong introduction and will save me some time. And the next thing I'm going to talk about is how sex differences play into changes in ion channel expression, and how that is in fact the underlying mechanism that elicits the greater vulnerability of women than men to torsade de pointe. And I'm going to give you some new data, some that hasn't been published yet, on demonstrations that there is a genomic regulation of ion channel expression by estrogen. And that estrogen, in fact, alters copy numbers of ion channels in a very heterogeneous and specialized way, and that, in fact, is what makes the difference in terms of eliciting the arrhythmia phenotype. And I may skip on this data. It has to do with what happens if you essentially remove the sexual hormone from an adult rabbit, and how that plays into the arrhythmogenic phenotype. Let's first, in terms of background, I think a lot of you already know that you have the QT interval and prolongation of QT interval, which is a completely descriptive phenomena, has a very severe and dangerous issue. And in terms of pharmacological studies, any drug, not necessarily cardiac drug, psychiatric drugs, antihistamines, antibiotics, have been shown to cause this kind of effect in the human EKG. The list, if I showed you a slide of that, would be absolutely enormous, and in actuality of all the number of drugs that cause long QT. The basis for long QT is not unique. There are many forms of long QT syndrome. LQT1, which is essentially a mutation of KV-LQT1, which encodes for the IKS current and reduces. So it's a loss of function of IKS. Long QT2, which is essentially a loss of function of the IKR current. LQT3 is the sodium window current increases, and so on. I'm going to focus here in LQT2, much as Bianca did, because not only is it the most common, but because, as Dennis Noble expressed in the very first lecture, the IKR current is perhaps one of the most sensitive currents because the hERG protein is very promiscuous and it interacts with so many drugs. And so it's a real health hazard and a health issue in terms of providing drug safety in virtually any drug that's being put in the market. What happens in the female heart? The female heart is quite different than the male heart. So in women, there's a much greater risk to drug-induced long QT1 and 2. Now, these are the only two that show this kind of differentiation between men and women. LQT3 is equally lethal to men and women. So we are focusing on these two because here we have a physiological model where normal ion channel expression has a very different outcome when you block one of these two currents. LQT2 is much more of a health hazard than LQT1 and is much more common, again, because of the promiscuity of the hERG channel. So these drugs elicit a type of arrhythmia called torsade de pointe that was first characterized by Dessertin in 1966 as an oscillatory waveform on the EKG. And boys less than 14 years old have a much greater risk of lethal arrhythmias in congenital form of the disease because you don't have too many drug-induced form. And this was essentially something that was reported only recently from the human registry data at Rochester. And the female gender in the adult shifts, and they then have the greater likelihood of the disease, of the arrhythmogenic phenotype. So previous studies had focused on how sex hormones affect ion channels, and they had mostly focused on the effects of estrogen reducing the IKR current a little bit on the IKS current. Much less was looked at in terms of the effects of sex hormones on calcium currents. So from this point, we give you this experiment. If you take a male adult rabbit, and here you have a mapping of action potential, so I'm just showing one of the action potentials from the surface of the heart, and you see a normal beat, beat to beat, and then you add an IKR blocker. E431 is a pure blocker of IKR, highly selective, and at a concentration of half a macromolar, the action potential durations become very long. You can see these go from 300 milliseconds to over a second, and you see a few humps here and there, but there's no arrhythmia. If you do the same thing with a female adult rabbit, the moment you add E431, this builds into a torsade de pointe. It's a classic oscillatory arrhythmia, and it takes about 5 minutes to occur. Now, if you shift and do the same experiments with prepubertals, in the prepubertal male, you have the classic torsade de pointe. In the prepubertal female, you don't have the torsade de pointe. So there is a shift that occurs at adolescence. In the rabbit, this is day 49 and up, where the surge of estrogen and testosterone changes the landscape of the arrhythmia. Well, in the past, people have argued that the difference between male and female is that the female action potential is longer, it has less of a repolarization reserve, and therefore is more arrhythmogenic. But if you look at action potential durations as a function of cycle length, in the prepubertal adolescent female rabbit, the action potential durations are indeed longer than the male, so action potential duration is not a predictor of the arrhythmia phenotype. This happens to be also the case in the human population. There's a wide range of action potential durations or QT intervals that are observed, and some are symptomatic. They have long action potential durations and they have highly vulnerable to torsade de pointe, and others aren't. And the reverse can happen. They have relatively short action potential durations, well within the range of norm, yet they're very vulnerable to these arrhythmias. So the question is, what's the mechanism underlying these differences? The first thing is IKR is not enough to explain this, and more we now started to look at calcium handling. There is a general consensus in the literature that torsade de pointe is triggered and elicited by early after depolarizations that fire in the plateau phase of the action potential. These are depolarization that go one or two shots, then you get salvos, and you progress from that point on. The controversy is whether or not early after depolarization are caused by intracellular calcium oscillations or are caused by spontaneous reactivation of the L-type calcium current during the plateau phase. So why does QT prolongation elicit EADs in some cases, in females but not males, and why is there a reversal? This is an example of action potentials. This is the control, and now you add dofetilide, another IKR, very selective IKR blocker, and one of the things you notice is that even before there are any early after depolarizations, you start to see oscillations in the calcium This is essentially a system where we have two cameras. We stain the heart with a voltage sensor and a calcium indicator dye. And in the past, in 2000, we had validated the interpretation of these signals, that there's no crosstalk. But you can see very clearly the behavior of the calcium intracellular transients, very different from the voltage, where you have these calcium handling problems, in this case, long before you have electrical events. So we started to do an analysis. This is essentially a schematics of a rabbit heart, and this is sort of the area that we're analyzing, where each box here represents recording sites. So when we examine essentially where is the initiation of the first EADs that occur, they all tend to be clustered near the base of the heart, and this is adult female rabbit hearts. And these are different experiments. These are different locations for different rabbit hearts, and there is very little here. So what do I mean when I say the base of the left ventricle? I mean basically this region of the heart where you have the largest diameter, and hence the location. And this is reversed in prepubertal. So we isolate cells from male and female hearts, and we examine cells that are from the base and of male and a female myocyte, and you can see that the L-type calcium current is markedly and statistically significantly longer in the adult female. At the apex, they're basically the same. The current to voltage, the current to voltage relationships are the same. Now, if you go to the prepubertal, this is reversed at the base, where the female is now lower than that of the male. This is accompanied by protein analysis, where you have differences, marked differences in Western blots for different animals, the female, male, apex, female, apex, male. In the prepubertals, this is reversed. So if you have an increase in influx, you must have an increase in efflux. So NCX is also regulated, and it's much in the same way. If you inhibit the NCX current with KBR 7943, you can stop the arrhythmias. So both L current and the sodium-calcium exchange current are modulated. If you do histology, you can show that in the prepubertal female, you have much less NCX than in the male, and it goes across with higher expressions in the base than in the apex, and a uniform expression in the female. Western blots concur that this is the case for NCX, and this is essentially a repeat with adult Westerns, and oops. And now we analyze the current to voltage relationships of the NCX current. This is the nickel sensitive current, and you can show that the female NCX current is higher than the base. They are the same at the apex. In this is prepubertal, this is adult. And now, how does this work? If you analyze the time course of the changes in the current as a function of time, you can culture the cells for 24 hours, 48 hours, and 72 hours. And what happens is that if you look at the male current is low and it stays low, and the female NCX current is high. After 48 hours, it becomes the same as the male, and then they're the same again. So as you culture the cells as a function of time, the two become identical. So basically what happens is that the currents are essentially upregulated by estrogen, and if you remove estrogen in the medium where you grow the cells, you see the decrease in current amplitudes. And this is, again, another way that this phenomena works at the base, but it doesn't work at the apex. The apex isn't regulated. So let me go on a little faster here. What is the mechanism? The mechanism is through the alpha subunit of the estrogen receptor, because if you apply estrogen plus the antagonist, you knock out the effect, and you essentially can show that the alpha subunit of the estrogen receptor, because there's an alpha and a beta, but the alpha is sufficient to explain the mechanism. And you can also use an antagonist, and here it is, the fulvestrant, that controls this effect. The L current, likewise, if you do this experiment control plus estrogen, this is the peak L current increases markedly. I don't think you can see these cells before and after. And isolated cells show the same phenomena. Here you have a control action potential with the fatal light, a cell isolated from the female base. At the apex, you don't see any humps. You see the large prolongation, but no EADs. With estrogen in the bath for 2 days, then you see the EADs. And if you block it with the alpha ER, you block the EADs. These effects, in conclusion, are not just for the rabbit. In the human heart, Verkerk showed that in female myocytes, you also have a difference in L current, and it's about 10%. It wasn't statistically significant, but there was a wide swath of types of hearts that he was able to get. But the female heart with the elevated L current shows the EAD in this case, and the male didn't. So let me quickly say a word about what the significance is. Repeatedly, you hear clinicians say, I have an individual who has no electrical disease. He has asymptomatic despite having a congenital form of LQT. Lives to the age of 45 and no treatment necessary. Then he dies abruptly. And you say, why did he die on Wednesday and not on Tuesday? So some things change in the environment. And as we age, our levels of sex hormones change. Hypertrophy changes through other forms of remodeling in terms of ITO currents. And nearly every form of heart failure, whether model or human data, shows that NCX is elevated. And so this plays an impact on trying to predict who is going to have a severe arrhythmia triggered by EADs or not. Thank you. I'm sorry.
Maybe one quick question.
What I would be interested in is the connection between the estrogen receptor and the upregulation of the calcium and the NCX. What is the pathway for this?
Right, so this is a wonderful question, and we are working on this very issue. There are two forms of genomic regulation that are known to happen. One is a classical, and one is a non-classical. The non-classical is an effect of estrogen receptor complex having an effect on kinases. And the activation of kinases has an effect on transcriptional, nuclear suppressors and activators. The other form is that the estrogen receptor complex binds to estrogen receptor response elements on the DNA and controls translation. So we now are looking into this very issue and find that certain kinases are upregulated at the base compared to the apex. So we have a huge benefit of comparing in the same heart the two cell types. So it seems that you upregulate the L currents, but you downregulate the K currents. That is probably by classical downregulation of the K currents and a non-classical approach of kinases for the calcium currents.